So the second part of the basic mouse genetics, I wanted to introduce genetic recombination. Mendel, who was around from the 1820s to the 1880s, who was a priest, but also was also interested in inheritance, and obviously did his experiments with pea plants, but following inheritance of traits in peas and in pea plants. And he developed a number of laws called Mendel's laws, which we know as Mendel's laws. The law of segregation, so diploid individuals, so individuals with 2 copies of each chromosome, or two copies of each genetic element, produce haploid gametes, so the gametes that have 1 set of those elements. 2 copies of each gene segregate from each other into different gamete cells, so in meiosis and in gamete generation, so in mammal sperm and egg. The 2 copies of each gene, which form the pair, will separate into separate gametes. And the child of one of those conceptions will receive either allele with equal probability. So a 50% probability, or 0.5 if you like. So there's a 50% chance of inheriting a particular allele from each parent, and a 50% chance of either inheriting one or the other from each parent. And then there's the law of independent assortment. So 2 loci, which we can call A and B, will independently assort during gamete formation. And then the products of those alleles will occur at lower frequencies. So if a child can receive either allele with the equal probability of 0.5, then that's 0.5 times 0.5 gives you 0.25. So if you inherit A and B, or different allelic version of A or B, or heterozygous, and obviously this only applies to unlinked loci. So you only get random assortment when the loci are unlinked, so they're not constrained by being on the same chromosome, for example. We'll come to that in a bit more detail in a minute. So this was forgotten for a long time, Mendel's studies, but rediscovered. And the first Mendelian inheritance in mammals was following the inheritance of coat colors, which are obviously easy to follow and look at, in 1902. This is a paper in 1902, Lucy and Queno. In 1903, William Castle, who we've already heard about at the Bussy Institute, published a paper on albino coat color genetics in mice. So these are the first genetic mapping, genetic studies of inheritance in mice. So in meiosis, so in generating gametes, which is generated by a process called meiosis, there's a stage of this meiosis process that takes a diploid cell and gives you haploid gametes, where the chromosomes pairs are aligned and you get crossing over. And it's this crossing over that leads to, which is recombination. So the chromosomes recombine. So you've got 2 homologs of the pair. They form a chiasma, and there's a recombination event. So they actually swap bits of DNA. So, and then you get 2 separate chromosomes that have swapped DNA. So that gives you that's the basis of it's that event that allows us to do genetic mapping at a subchromosomal level. So we can follow the segregation of unlinked genes that are on different chromosomes. But this allows us to go even finer because the DNA swaps between chromosomes as an event during meiosis. So it gives us more power to map, which I'll explain in a bit more detail shortly. So the other aspect, the other features of meiosis are independent assortment. So you reassort all the chromosomes. There are 20 pairs of chromosomes in the mouse assorting independently. That gives you 2 times 10 to the 20 combinations, a million combinations. You get random fertilization. So you've got 2 times 10 to the 20 times 2 times 10 to the 20 combinations. So it's a huge amount of potential for reassorting and for giving us variation that evolutionary forces can act on, for example. So not only we get the recombination, we get the independent assortment of alleles, we can generate individuals with quite a lot of, if the polymorphism's there in the first place, the potential is to generate large numbers of different permutations of those polymorphisms on which evolution can act. So it's a very powerful process, and it's the engine of genetics and evolution. This is what gives us the variation and the permutations of variation that evolution can select. Should they provide advantage or disadvantage? So, as I said, on average, there's 1 recombination event per chromosome meiosis. The homologous chromosomes are held together prior to anaphase, the first meiotic recombination by Chiasmata. So you can see visible evidence karyotypically of these events, which I'll show you on the next slide. So on average, this occurs once per chromosome. So there's the opportunity to swap DNA once per chromosome per meiosis. And there's a bit of terminology here. So Centimorgan is a genetic map unit of length. And you heard about those in the other talk when I was talking about the length of chromosomes. So 2 loci are said to be 1 centimorgan apart if recombination events are found in 1% of the meiosis. So if you've got 2 visible traits that you can follow in your mice, and you do a breeding experiment, and they separate in 1% of those individuals, you'd say they were 1 centimorgan apart. And this is old data, but it's the data that these numbers are based on. These are different strains or the different groups, actually, working with different mice and measuring chiasmata. And so, 50 to 80% of the chiasmata are seen as single events. But there are more frequent events as well. So there may be 2, there may be 3, there may be 4. So you may get double or triple recombinants, but they get rarer and rarer. So 1 is the average. So genetic mapping. So these recombination events, these recombinations between the chromosome pairs are relatively random. They're not totally random. And the likelihood of recombining and separating two markers on the DNA, or phenotypic markers that correspond to changes in the DNA, increases the further they are apart. So if you've got a chromosome and there's a recombination event at random, once per meiosis for that chromosome, and you've got 2 markers, if they're further apart, there's more chance of a recombination event occurring between them if it happens once per chromosome. But if they're really close together, the chances of that recombination event occurring is lower. So if you want to separate, there's 2 things that you can see phenotypically, for example, or genotypically if you've got a DNA marker, the chances of them being separated when they're close together is lower than when they're further apart. I think that makes sense, doesn't it? Because if there's an average of 1 recombination per chromosome, and they occur at random, then the chances of 2 things close together being separated is lower than if they're much further apart. And that's the basis, really, of being able to map. If they're close together, they're more likely to stay together during meiosis. If they're further apart, they're more likely to separate. So and two markers, as we said, on two different chromosomes will segregate independently. So they'll be transmitted 50% of the time to a gamete. So one of the first genetic maps was ordering 6 sex-linked factors in Drosophila in 1913, and then the mouse shortly afterwards, pink-eye dilution. So these are some of the first genetic maps based on meiotic recombination. And they're in the Silver book, Lee Silver's book. So I've talked about phenotypes, but obviously, in genetic mapping experiments, we want to follow the segregation of DNA and look at the segregation of that DNA because we want to know what the genes are. We want to know what the function of the genes are. So we want to follow the segregation of DNA with phenotypes that we can measure. So it's necessary to be able to distinguish the DNA of the parents or the DNA of those two chromosomes that are going to undergo recombination events. So we need to be able to tell the difference, or we won't be able to see the difference. We won't be able to see these recombination events if we haven't got some way of telling the difference. And the way of doing this is to look at DNA polymorphisms, so single nucleotide polymorphisms, so-called SNPs. And these SNPs distinguish different. So if you compare different inbred strains, you'll find they have different patterns of SNPs. So if you cross two different inbred strains, you could follow the segregation of the DNA from those inbred strains in their offspring, because their DNA has these polymorphisms in it, these SNPs. So they're not necessarily encoding DNA. They're across the whole genome, and there are huge numbers of them. And if you want to identify some SNPs to follow, you can look at the Sanger databases and compare strains. So they have search tools, so you can put up, search the 2 strains that you're interested in, at least the ones that have been genotyped and sequenced. And you don't have to do this in-house. It might be cheaper for you, in fact, to send your DNA away in a plate, in a microtiter plate, and get it genotyped commercially, rather than set up all the technology needed to do this. It's much easier and probably cheaper, if you counted all the other costs, to send these things away. So if you want a genetic map location, you need to have some mice that differ, 2 populations of mice that differ in phenotype and genotype. You need to generate a population, cross them together, generate offspring, phenotype those mice for your trait, blood pressure, glucose, obesity, whatever, behavior. And then, having got the phenotypes, to genotype the mice with genetic markers of known location. So we know where all the polymorphisms are in the mouse genome because they're mapped in databases now. So you can then, if you make DNA from those mice, you genotype that DNA, and then you can ask the question, what DNA, which bits of DNA have been inherited with your phenotype that you're measuring. So you correlate genotype and phenotype, and ask which genotype pairs with the phenotype more often than expected by chance. And from that, you can calculate your map location. Does that make sense? So those are the basics of genetic mapping: having polymorphism, exploiting recombinations, being able to measure phenotypes, get the genotypes, compare the 2, and identify which bits of DNA are going more frequently with your trait of interest. So, it follows that you could work out the genetic length for the mouse genome, and this is around 1,300 Centimorgans. How are we doing for time? So genetic mapping is all about collecting recombination events, because we want the more recombination events you've got, the better the map you can make, because markers close together don't separate as frequently as markers further apart. So this is going to give you the resolution for your genetic map. And you can collect recombination events in a number of ways. So back-crossing, intercrossing, you can use recombinant inbred strains. There's the collaborative cross, HS stocks, and outbred stocks, which we sort of alluded to. And I'll go through these in a little bit more detail. So back-cross mapping. So in a back-cross, you want to, if, for example, we want to map a dominant trait, and say our black 6J mice have got a trait that we're interested in. We don't have to know if it's dominant at this stage, but we cross it to a different strain of mouse, C3H, and we generate an F1. So they inherit one set of chromosomes from black 6. Each mouse has one set of chromosomes from black 6J and one set of chromosomes from C3H in an F1 animal, or a G1. Sometimes you see it called a G1. And then if we back-cross that to a C3H, then we'll generate a back-cross 1 generation, or a G2. And these F1s, when they generate their gametes to make these back-cross 1 mice, will undergo meiosis as part of that process. So there will be recombination events. So your back-cross 1 mice will contain recombination events from this F1 individual. And so they'll have potentially a recombined chromosome opposite a C3H chromosome. So this is fine for dominant traits. So you'd measure your phenotypes in this generation. You genotype the DNA. You've got polymorphisms between C3H and black 6. Follow the segregation in that back-cross population. The number of these back-cross mice, how many you have in your cohort, determines the number of recombination events, which determines the resolution of your map. So, this is a bit extreme, but 1,000 back-cross 1 mice yields at 95% confidence a resolution of about 0.3 centimorgans, or 500 kb. So if they were very close together, so if you want to know exactly where it is sort of gene-wise, you wanted a resolution of 500 kb, you might need 1,000 back-cross 1s. But if you just want to know a region of a chromosome, you don't need as many as that. That's a lot. So you can the resolution of your genetic map. So if you have a trait and you don't know what the gene is, depends on the number of recombination events that you've managed to collect. So this is just more visually. The recombination events, and then back-crossing so you've got a C3H chromosome and a recombinant chromosome, for example. So this is just a bit of old but real back-cross data. This is actually a glucose trait. So you can use software to do these analyses, but you can also manually look at your genotypes as well. So this is different genetic markers across a chromosome. So each column is a different genetic marker. These are the traits here. The ones in red are high, and the ones in white are normal values. And you can see and each one is an individual. Each line is a back-cross 1 individual. Okay. So if we look at this individual, all its DNA is heterozygous. So it's got black 6 and C3H DNA. The same for this one, although here there's been a recombination event and it's got just C3H DNA. Here there's another recombination event in these 2 different individuals, somewhere between these 2 markers. So these markers are in order along the chromosome. So it's a picture of the chromosome, and there's a recombination event here. So we can say, and these mice don't have the trait, some of them have recombination events and bits of black 6 DNA, but we can say fairly confidently then that the gene is probably in here somewhere, and these recombination events would say it's to this side or to this side. So it's in this middle bit. So this is a bit that's in common. These mice, it's not going to be here because these mice have this heterozygous DNA but don't have the trait. So that, hopefully that isn't too confusing, but that's one way of looking at your data. And there's a chromosome where there is no linkage. So the trait's not on this chromosome, it's all C3H DNA. These mice have the trait, but on this particular bit of the chromosome, there's nothing. There's no inheritance of black 6 DNA. And we're looking for a gene that came from black 6 that's on this backcross background. So we're looking for mice that are heterozygous. So you can treat these as quantitative traits and use mapping software, and these use LOD scores, so the probability of obtaining a set of observations. So I won't go into that in too much detail because we're running out of time, but a lot of 3 or more is generally taken to indicate that 2 gene loci are close to each other. So your phenotype you could call a phenotype close to a genetic marker. So not necessarily just genes, but a phenotype and a gene. There are a number of different packages, and RQTL is one of them. It uses the R package, which is freely available. It's not particularly user-friendly, but if you take the time and effort, I can see Ines saying it. Well, we send our students on an R course, and some of them really like it, and some of them hate it. But the advantage of it is that it's freely available, and it's written by statisticians, and there are modules, plug-in modules like RQTL. So, and there are books on using QTL mapping in R, but it's a serious investment of effort, but well worth it. So this is just another example of backcross mapping where we mapped a glucose trait here to chromosome, well, to the glucokinase gene, in fact. So this is a MODI model that we published a while ago now, but it's just an illustrative example. So this is using some mapping software. So you can see that we have different traits in different colors here. So, the pink is T60 in a glucose tolerance test, blood glucose concentrations. And we can see that trait has a LOD score of greater than 3 in this region, which suggests this, and we found a mutation in the gene. So that just sort of illustrates how you might use this sort of approach. So you'd map a location, you'd look up the list of genes, because this is all available now through databases like Ensemble. You look at all the genes in the region. You might select some candidates for sequencing to look for mutations. Or increasingly these days, we just send the DNA for whole genome sequencing. But you need to know where your gene is, because you'll get a large amount of sequence data back, and but you'll only look at the bit that corresponds to that bit of the chromosome. So you'd look at all, if you went for whole genome sequencing, you'd look at all the mutations in this region here, and then have to do more functional downstream work to validate those. As I said, sequencing may be more cost effective. Depends on where your major costs are, but for us, keeping mice is quite expensive. So there's a balance between breeding more mice and collecting more recombinations and going straight for sequencing, and then for some sort of functional demonstration that we've got the right mutation and gene. So there are different sorts of mapping results. So I've talked about backcrosses, but obviously a backcross won't work for a recessive trait where you need to have homozygosity for a mutation. If you've got a genetically complex trait, so you've got several genes, one gene or more than one gene, 2 genes, more genes, segregating that control a trait, which is quite likely for a trait like blood glucose is not going to be controlled just by one gene necessarily. It's going to be a complex trait. So, and your choice of mapping resource is also influenced by and the sizes of the cohorts that you're going to generate is also influenced by the size of the effect that you're trying to measure and how much variance is explained by the genetics. So there's always going to be some environmental noise in there as well, and the accuracy of your test. So another approach is F2 mapping of a QTL. So quantitative trait so this is QTL mapping, quantitative trait loci. So traits with numerical values, often with complex genetics. Loci may interact, loci may be small effect. Trait sizes may vary with genetic context. They can be challenging to clone these genes, but with next generation sequencing, that's got a lot better. So as I've alluded to, we mapped a glucose intolerance trait in 6J mice. These are C57 black 6J mice in a glucose tolerance test. So the mice are fasted, you measure the blood glucose, you give them a glucose challenge so their blood glucose goes up and then comes down over a couple of hours. And you can see black 6 is different to, say, C3H in the pink. So there's a clear difference in glucose tolerance at 12 weeks of age. They show differences in insulin secretion. So black 6 mice secrete less insulin during a 30-minute glucose tolerance test than C3H mice, so they have lower insulin secretion. We showed that the KATP channels showed impaired glucose sensitivity, suggesting that the mutation was upstream of channel closure. So in beta cells, the KATP channels have to close before you get insulin secretion. So we carried out intercross mapping, F2 mapping. So in this regime, you cross a black 6 mice with a C3H mouse, you get an F1. You intercross those F1s. And this mixes the DNA, but it also means that we have 2 visible meioses in these offspring for each chromosome. So you've got more recombination per chromosome in this strategy because you're collecting the recombinants from 2 different chromosomes. So we have DNA now in this F2 or G2 population that is homozygous for some black 6 alleles, homozygous for some C3H alleles, heterozygous for the 2. And in that population, you can follow the mapping, follow your trait, which in our case was glucose tolerance, which we did. We had 260 F2s. We did a relatively modest, by today's standards, genome scan with a different sort of genetic marker. We found 3 linkages on 9, 11, and 13. There was a strong candidate on chromosome 13. So here there's chromosome 9. So you saw these sort of QTL mapping output plots for different traits. Here's 30-minute insulin. This is T0 glucose. And this is 160. This is 60. T60 glucose, and there's this strong peak on chromosome 13. So let's cut through to this. So we were able to identify a gene underlying that trait. So, but this was a complex trait. So the trait was influenced by at least 3 genes, and that didn't explain all the variants anyway of the trait. So epistasis is where the effects of one gene are modified by another. There are gene interactions, there are modifiers, there's incomplete penetrance. So you might inherit a gene, but you won't see a phenotypic consequence. And there are stochastic elements as well. So if you're looking at a quantitative trait, you'll quite often find that the trait distributions will overlap. So there'll be a group of animals in the middle that you perhaps can't distinguish because they might be high normals or they might high wild types, or they might be low mutants because it's a quantitative trait and it varies for all sorts of reasons, including random ones. You should also be aware of imprinting, beware of parent of origin. So that's another reason for keeping good records and knowing what you're breeding and where alleles are coming from, because you may come across genes that are imprinted. So the imprinted genes are those genes which are only expressed from one chromosome, so the maternal copy of a chromosome, for example, or the paternal copy of a chromosome. Okay, I'll try and speed up a bit. Recombinant inbred strains are another choice of another genetic mapping tool. These were generated from crossing 2 strains together and then generating F2s and then interbreeding those by brother-sister mating. So you end up with a set of lines where the loci are fixed, but you've trapped all these recombination events that have occurred in the breeding strategy. So each line has a unique set of recombined alleles from the original parents. So there's some advantages of that, because you can get those lines, and they're genetically fixed. They're a renewable resource. You can measure multiple phenotypes. You can measure lots of phenotypes in lots of different mice. So you could, for each line, you could measure a number of different individuals who will be identical to each other. So if you've got if you've got a certain amount of variance in your trait, you have the advantage of being able to measure it multiple times in one set of, for each set of recombined alleles. So each one of these lines is separated, and you can have multiple individuals in each one. But so you compare across them, but you can also get data within a line. Genetic map data is already available, so people have already genotyped them, so you don't have to do the genotyping, and the maps are made. The disadvantage is that there are limited combinations available, and they're small panels, so the resolution's lower. But it's worth considering, and they're out there still. You can buy them from Jack's. Consomics and consomics. So consomics are chromosome substitution strains. So there are mice available where a single whole chromosome has been moved onto a different genetic background. So if you're interested in a particular chromosome or traits that are on that chromosome, you can look at it on a different genetic background. So they'll have chromosomes, all the other chromosomes will be homogeneous from another strain, but you'll have your one chromosome of interest. And then you can use that strain then to do to do genetic mapping experiments. So you can break that chromosome up once you've got your trait information and generate lots of collect recombinations on that chromosome, on that clean background. Obviously, multiple QTLs on a single chromosome. But there are limited panels available. Congenics, this is a little bit different. So if you've identified a region of interest for a trait, you can repeatedly backcross. So the colors aren't very strong on here, but so say you've got a trait controlled by a gene in here, you can repeatedly backcross onto a selected background to remove other bits of DNA, and then you phenotype these individual congenics. So they have a clean genetic background, but they have small segments of DNA, and recombination events make these smaller and smaller. And if you collect different individuals, then you have different breaks in different regions. So you can get a line that retains a particular piece of DNA that you can phenotype and further investigate. And if you have a selection of these, you can work out where your gene might be. So you can see that there's a recombination event. So if the mouse carrying this chromosome has the trait, and these other mice do, then you would infer that the gene was just here because this is the only bit of DNA that they have in common, if you look across. So that's this. If this is the chromosome with the mutation on it, this animal has it, this animal has it, this animal, this animal. But then it can't be here because this one doesn't have it and that one doesn't have it. There are high-resolution mapping strains as well. So the heterogeneous stock, the HS stocks, for example, and outbreds to some outbreds as well. So these are using genome-wide association type strategies in the mouse. And there's a good review by Flint there in 2012. So they were made from classic inbred strains, and are a combination. I'll not put it on there. That's on the next one. They were made from 8 different inbred strains. The collaborative cross was. And multiple intercrossing generations. So there's large numbers of recombinations, and there's also public sets of genotypes for each strain. So you can do a fairly high-resolution mapping experiment using these strains, if you import them, that is. And HS stocks, so this is just a sort of graphical illustration of HS stocks, but again, 8 inbred strain cross. So you've got lots of bits of DNA from the 8 strains on each pair of chromosomes. So it gives you quite a high genetic resolution. You've got lots of genetic variation from the 8 strains. You've got lots of recombination events, and you've got lots of individuals. Outbred stocks is another aspect of the same idea. So you've got large numbers of recombinations and large amounts of genetic variability. So I think I'll just skip through to near the end now. So this is sort of practical consideration. So when you're doing genetic mapping experiments, check that you're getting the genotypes that you expect, and test using chi-squared tests. If things are not being inherited as expected, then there may be problems with the genotyping, or maybe a class of mice is not being born because of embryonic lethality, for example. So question your data. So I'm saying collect the data, question it, and ask whether or not it's you're getting Mendelian inheritance or not. So genetic background, as I've told you repeatedly, strongly affects traits. Test different genetic backgrounds if you can. Know what background you're on, and clean it up by backcrossing or outcrossing or remaking the mutation, as Ken suggested. Use the appropriate controls. Use glutamate controls where you can. They're the best control. And think about cohort numbers and power. Because inbred strains do differ. So this is this paper I told you about in genome biology, comparing genotype and phenotype differences between 2 strains that are very similar, didn't diverge that long ago, 6J and 6N. But there are 34 SNPs encoding regions, 32 missense mutations between these 2 strains, 2 frame shifts, a nonsense splice mutation, deletion in the gene that we mapped. So use the right controls as best as you can. You need to think about cohort numbers as well. You need to have a properly powered experiment. You need to, as people have already said in the audience, you need to consider age, sex, and environment that these mice are in. It helps to have, if you have experience with your assay and numerical values like quantitative traits for glucose, it's useful in power calculations to know what sort of variance you get on a trait. Yeah, and the mapping power differs by the type of cross. I'm just going to skip over that. So the power to detect a trait depends on the variance of that trait. So if you know the mean of your trait value across a population of, let's say, your inbred strain, and you know the standard deviation, it's easy to use software like R or GraphPad StatMate to calculate power. So, for example, in this glucose tolerance test at 12 weeks, and where the standard deviation was 4.36 millimolar, and the difference between the group averages, so wild types versus heterozygotes, that we wanted to detect was 5.9 millimolar. Using this sort of software, you can determine that you need 16 wild types and 16 mutants to have a 95% confidence with a p-value of 0.05 of being able to detect that sort of difference in your experiment. So there's no point, you need to guard against your experiments being underpowered. Obviously, you don't want to use more animals than are necessary, but you don't want an underpowered experiment where you might make an observation by chance, which is wrong. So, and you will increasingly be asked in publications to provide this sort of information. What's the power of your experiment? Yeah. The NC3R's website is quite useful for looking at. It's a UK organisation, but they have lots of useful advice on power calculations and design of experiments, so it's worth a look. So why is this stuff important? Well, there are reproducibility issues across science really, but also in mouse work as well. And there have been a number of papers like Make Mouse Studies Work. You need to do experiments that are going to generate robust conclusions and that are going to be reproducible. So you need to think about all of these things that we've been talking about: background, cohort size, sex, environment. You need to do properly designed and planned experiments. And there's this thing called the ARRIVE or ARRIVE guidelines, I'm not quite sure which, which provides useful guidelines on these sorts of variables and on reporting these variables in publications. So trying to standardize things so that we get better reproducibility. So you need to be aware of these because lots of journals are now asking for you to be compliant with these. So there's no point in doing an experiment and then realize you've missed something out because you might not get your paper accepted if you don't meet the guidelines or can explain why you don't meet the guidelines. So, be aware of these guidelines. It's useful information and advice. So the mouse is a very powerful tool for genetic studies. It's a great model system. There are fantastic resources out there, publicly available. We can manipulate the mouse genome. There are great techniques coming along. But planning and understanding what you're doing is quite important to saving money and getting reproducible results. So I think you're going to hear about international, so this is just some trends that I stuck up. International Mouse Phenotyping Consortium you're going to hear about. Whole genome sequencing has got a lot cheaper. There are these big maps that ENCODE and other organizations are doing, providing publicly available data on DNA methylation maps, histone methylation maps, chromatin modification maps. There's useful data for your experiments potentially. This is just my own pet thing, that trying to understand which genes underlie genome-wide association, study traits in human using the mouse as a model. And I guess the big thing at the moment is CRISPR-Cas9 for genome editing, so technology which I think we're going to hear about this week, but hopefully we are, for editing the mouse genome, so quicker potentially than conventional technologies, and quicker and maybe cheaper. So that's it. Thank you very much. I'll be over.